no matter the level of scientists that you have, how polished they are, how trained they are, or how green they are, it's at the limit of the method at the end of the day. If you have an efficient method, it's going to be easier to bring someone in no matter their experience and train them on it.
Welcome to another episode of the Omni Pod, where we're enabling conversations between life science thought leaders to help you get the most out of your preclinical research.
Today, we're talking about onboarding new lab staff without losing data quality.
I'm Bill Schick, Fractional CMO, and today I'm speaking with Gabby Ryan, a Senior Applications Specialist at Omni International. Tell us about the first time you worked in a lab outside of university.
Yeah, great question. So I kind of have to think back here. I think the first time I was in a lab, you're not going to believe it, was actually in the Omni lab. I sort of am not your typical academic or university trained scientist. I quickly came out of university and found an opportunity at Omni. And the first time I was in the lab was here at Omni. So I was blessed, I think, with sort of a non-traditional lab experience. Being in an industry lab, you sort of have different goals and different metrics to hit, whether it's QC testing, product testing for new product introductions, and application testing as well to support customers. So it really is a unique experience being in an industry lab, and being able to support customers, I think, was one of the things that interested me the most. And creating custom solutions for them in the lab was one of the things that made me fall in love with Omni, because being my background is in medical science and in biology. So our customers do anything and everything. We always say they process everything from bones to bacteria. So, and everything in between. Like, I remember we had a customer one time that wanted to grind up PC boards for metal testing, and coming from a medical background, I was like, whoa, that is definitely something you don't see every day, and keeps you on your toes, being in the Omni lab. And it really just broadens the horizons. You're not working with the same things every day. So I would say it was a general positive experience, and definitely gave me a taste of, what working in this industry can be like, and especially supporting customers was definitely a high point for me, and still is.
Yeah, I think that's great. So one of the things that you've really gotten out of the job is seeing a lot of different sample types come through, and how different methods and protocols for customers come in.
Yeah, exactly. And I think, you kind of hit on it. Customers come from, and scientists come from many different backgrounds, and they're working in different labs, and they have different needs and different things that are important to them and to their method. And they can come to us with a variety of contexts, as I like to say. And, what they come to us with is generally, I have a method. It does what it needs to do, but it can be better. Whether that's, a customer coming, they're doing a mortar and pestle method, so they're pretty hands-on in the lab when they're preparing samples. And right, that works for 5, 10, 15 samples a day. But I don't know, if you ask me, I don't want to be at the bench with a mortar and pestle, grinding up manually samples every day. And so when they scale up, it doesn't translate. That mortar and pestle is left in the dust, and they come to us wanting a better way. And we sometimes take that to the lab. If a customer is super, they have a super novel sample type. I can think back to a customer who was processing beluga whale blubber, and they were doing some endocrine disruption testing. And oftentimes we'll even, again, take it to that degree where a customer is sending us a sample, and we're coming up with a method for them, really, really hands-on. And I think that's one of the things that sets us apart, is our ability to really go to the extra mile for the customer and create the method for them, show them a little bit of proof of concept data, show them a homogenate that looks good to them and what they were achieving before, to be able to, serve as that proof of concept, serve as that checkbox that, hey, this bead disruptor, this homogenizer is going to do what I need it to do. And at the end of the day, it's going to give me the efficiency that I need. I think that's the big thing, and the time savings as well. Because I remember the whale skin. I'm pretty sure we processed that in only, I think, a few minutes, and the customer was extremely happy with that. And they ended up, that's just the first step. Because it's a tip of the iceberg. Because once you realize the power of, let's say, the bead disruptor elite, you start, as a scientist, you're like, oh, what about this project? What about this project? What about this project? And so it just sort of compounds the effect of it and the efficiency that it has.
That's great. That's great. On the topic of today, when new staff first come into the research environment, what are some of the initial mistakes that, people often see that just kind of the new person does?
Yeah, that's another great one. So I think, in my opinion, I think no matter no matter the level of scientist that you have, whether it's someone who is, never picked up a pipetter before, or you have a PhD level scientist who's completed, 10 papers and they have all this, wonderful research background, I think the efficiency in the lab that you see and the ability for someone new to come in and pick up what you've already done is at the limit of the method. So if your method isn't efficient, if your method is relying on a lot of different parts that are less than robust, just to be generic, then doesn't matter how good your scientist is, how polished they are, how trained they are, or how green they are, how inexperienced they are. The method is going to suffer in different ways, obviously, I think. But it's at the limit of the method at the end of the day. If you have an efficient method, it's going to be easier to bring someone in, no matter their experience, and train them on it, than, if you had a subpar method.
So give me an example of each. So talk a little bit about what maybe an example of an inefficient method looks like, and then maybe what an efficient method looks like and why that matters.
Sure. Yeah. Another great question. So I'm going to use mortar and pestle. I think it's a great industry homogenization method that we see. Mortar and pestle is a great industry method that I think a lot of people use. It's cited in publications. You see it on ResearchGate all the time. It's tried and true, as you might say, and scaling that up is definitely where I think a bead reactor comes in. So let's walk that back. So an inefficient method, again, in my opinion, I think mortar and pestle is a great inefficient method, right? It can do what it needs to do for 5 or 10 samples, but if a lab is scaling up, let's say they validated initially with a mortar and pestle, it worked for their tissue samples initially. You're able to, it has some pros. You're able to, pour liquid nitrogen in and do like a nice cryo grind. But at the end of the day, when you're doing that full study and you're preparing 50, 100 samples a day, it just is not going to cut it. It would take you a whole week to mortar and pestle grind 100 samples. And I don't think anyone wants to do that. And that subpar method is a great segue into an efficient method, like a bead reactor homogenizer. So using the bead reactor Elite as an example and sort of walking through that scale up. So a scientist comes to us and they're like, hey, I need a bead reactor. And we're sort of walking through with them, getting the context, understanding what they're doing. And they're telling us, I'm using a mortar and pestle right now. It's great. It does what I need it to do. However, I'm now under the direction of needing to process 50 to 100 samples a week. And that's just not going to cut it for a mortar and pestle. And so they come to us and we're, walking through all the features of the bead reactor. It really is a shoe-in from the jump, and being able to take a sample that was previously processed on a mortar and pestle, usually a tissue sample is a common one that we see, and being able to fit that into a tube type on the Elite and process that tissue generally in seconds to minutes is what I like to say. The scale up is really seamless. There are a few things that I think, an efficient method has that can sometimes require a little bit of a frame adjustment for the customer, for the scientist. It's a little bit of a mindset shift. You have to sort of be open to changing the sample prep method and understanding that it's going to still give you the data quality that you need. Because at the end of the day, both methods, mortar and pestle and bead reactor, can give you the data quality that you need. But there's other ramifications around it, right? Like, how many samples are you able to process in, 10 minutes? Are you able to process your 50 to 100 samples in 10 minutes, or does it take you all day? So, I think those are some great high points between the two, and sort of comparing an inefficient to an efficient method.
That makes sense.
Yeah.
What I'm looking for what are mistakes that new people in the lab can make in the sample prep process. Obviously, not everybody makes every mistake, but what are some common mistakes that actually cause challenges down the road?
Yeah, I think one of the things that is at the top of my mind hearing that question is inconsistent or incomplete sample lysis. If and regardless of the reason, the effect of inconsistent sample processing, having tissue left over, or having sample that's unprocessed can drastically affect the downstream method, and that can be a common mistake. Again, for whatever reason, whether the sample prep method is inefficient or it was a user error, there could be many reasons. The next thing that also is at the forefront of my thought process here is the cleaning process that goes along with sample prep. Sometimes you're using stainless steel consumables or stainless steel accessories on a rotor stator that have to be taken apart and cleaned between samples. That opens up a lot of potential for manual handling and manual error as well. If the probe or the accessory to the homogenizer is not cleaned adequately, you're then bringing sample from the previous, or you're bringing sample material from the previous sample into that next sample and into samples after that, thus cross-contaminating for every sample down the line. Another way that a sample prep method can be subpar or inefficient, I think, is sample loss. A lot of times, again, that sort of ties in with the first one. If we aren't processing a tissue or a sample all the way, we're leaving sample on the table that won't be analyzed downstream because it didn't get broken up in the first step. And a reason why you can have sample left over on the front end in that first step can be an improper technique. It can be the fact that a scientist is grinding up, or a tech is grinding up that tissue sample in a mortar and pestle, and because the nature of a mortar and pestle is very manual and hands-on, good enough to me can be subpar to you, Bill. So that sort of manual nature can leave room or leave an opening for improper technique or improper handling, again, just because of the nature of the method. So I think those are a few. I think that was 4 things on the top of my head that sort of come to mind when you ask that question.
So from your perspective, what aspect of sample prep is the most challenging to master?
The most challenging part of sample prep to master, I think, is honestly not, it might not be what you think, but I think the most challenging part is actually opening up your mind to the fact that it doesn't have to be this way. And the challenging part, because in my head, there's nothing challenging about sample prep, right? So it's kind of hard for me to get into that space, but putting myself into the shoes of someone that maybe has a less efficient method or is working with a mortar and pestle, I think it's easy to get bogged down and easy to get used to the methods that we have. And I oftentimes think the hardest time, or the hardest thing, right, in jumping from a less efficient method to an efficient one, or even the hardest thing about sample prep in general, is just the fact that it doesn't have to be this way and it can change. And opening your mind to that is, I think, difficult, especially for a scientist, because we're used to following methods to a T. We're used to copying and pasting between day to day, experiment to experiment. So I think the hardest thing can be just opening your mind to the fact that it doesn't have to be this difficult, doesn't have to be this inefficient. You don't have to spend an hour even on the bench, manually processing your samples.
What about a case where maybe there are larger issues at play and changing part of my sample prep process or my protocol is more involved than me just opening up my mind?
It depends on how much it's, I guess, paining you, right? If it's a big enough problem, no one's going to care how long it takes or the paperwork that needs to be done, or I'm not going to say they don't care. Obviously, all of those paper trails and method updates and validations are important. But if the customer is, if their sample prep method is affecting them negatively enough, then they'll be happy to change. And they quite literally come to us like, Gabby, I need a better method. I need a better way. And we're here to support. So I think, I think that's a good answer there.
Well, you've probably seen a customer who has come to you with a challenge like that.
Yeah.
Like we have, our entire protocol, our entire system downstream is reliant on this, and we know there are better ways, but it's going to be tough for us to change.
When the customer comes to us, there are certain things that they'll say, right? Like, and it depends on the environment too. Like in a preclinical research environment where, those trails have to exist, those validations have to exist, a customer can be willing to change to a certain extent. Like if they're using a rotor stator homogenizer from another company and there's breaks and they need to buy a new one, there isn't a lot of, those are pretty one-to-one. There isn't a lot of change that's happening there besides the instrument that's doing the rotor stator homogenization itself, versus a customer that wants to switch from a well plate based homogenizer to a fully automated system. Those aren't necessarily completely one-to-one. So there's going to be a lot of validation on the customer's end that goes into onboarding that new method. So I'll say there's a little bit of flexibility when it comes to onboarding new methods, and it's not necessarily that there's an uphill battle, but if a method is one-to-one, like if a customer is using a bead disruptor from another company and it breaks down, or they have an issue with it and they need to buy a new one, a lot of times we can provide a one-to-one, maybe a few steps up in quality and efficiency for them. But because those two methods are so close or so similar in nature, there isn't a lot of hurdles or uphill battle for the customer.
So really, as a customer, I might think that it's going to be more difficult than it actually is.
Yeah, a lot of times. And I think when a customer realizes, and a lot of that I think comes with the context of being a scientist and being so locked into what you're doing and the method that you're doing. But once you get a taste of the other side, once you get a taste of efficiency, it's quite easy, right? I can't tell you how many times I have a customer that will adopt the bead mill in their lab. I go back to the whale blubber customer, and it opens the door and 6 other doors behind it. I get emails like, oh, I have a colleague down the lab that's processing, bacteria. What do I need? Can I use the bead kit that you gave me, or do I need another one? And then that cycle starts rolling and the wheels start turning, and it's just like, you just don't look back.
Right. Speaking about the common mistakes you see kind of early on in sample prep, how do those mistakes show up later on downstream in the data?
Yeah, I could talk for hours about this one. I'm going to use an example from a validation that I saw. So we recently developed an application for an immunoassay, and the previous method for Lysis using that immunoassay was a sonication method. And the nature of this immunoassay it's a cytokine release syndrome assay. So we're quantifying cytokines downstream of a sample lysis method. And the kit, it's a Legendplex kit from BioLegend actually, and their kit does a great job of quantifying. I believe it's a multiplex kit. It quantifies like 13 cytokines simultaneously, and it's a wonderful kit on the back end. But if the front end sample prep wasn't done correctly, we saw a drastic change in the cytokine profile that was seen on the back end compared to what Lysis method was used. For example, using a sonicator was not lysing the sample all the way, especially skin tissue, which can be notoriously hard to lyse. But even for traditional soft tissues like spleen, like lung, those tissues were homogenized in a very different way with the sonicator compared to the bead disruptor elite. And we quite literally saw cytokines present in the bead disruptor elite samples versus PBS controls. So we getting into sort of the meat of the study here, we essentially induced cytokine release syndrome in a mice model, and essentially what we're doing is inducing those cytokines to be produced so that we can measure them on the back end compared to like a negative control. And we were literally seeing cytokines show up in the bead disruptor samples versus in the sonicated samples, we did not see the same profile. And so it was really eye-opening to the fact that a sample lysis method can quite literally cause your data profile to change completely. And I think thinking back, right, like if we were just, two groups in industry labs working on an application, but it sort of made us think like, wow, a customer using a sonicator. And if we were in a true research setting, this would have been like wheels are falling off the bus here, because, the customer would have maybe looked at, is my mouse model not right? Do I need to dose more of my LPS to get more cytokine response? I mean, it really just opens up a time sink for the customer and for the scientist in the lab when things don't work out right, and you start having to question everything that happens ahead of getting that single data point. Another example that I will give is in bacteria samples, especially for metagenomic sequencing. Those bacteria can be notoriously tough to lyse. And if the lysis method is not par and it's not lysing all of the different bacteria within that sample, because not all bacteria are made equally, they all have different degrees of lysis efficiency that need to be there in order to lyse them. Gram positives are different than gram negatives, and there's a whole rabbit hole we can go down there. But if we're not lysing all of those bacteria, then your metagenomic sequencing profile can look drastically different compared to if you were. And so you can just see how that would compound the effect of having a subpar lysis method can literally change the cytokine profile. It can change the metagenomics profile that you see on the back end if you're not lysing those samples accordingly.
Do you have any suggestions for reducing the variability with new hires, with new scientists in the lab?
Yeah, it really, I'm going to answer this question from the beginning of an experiment, which is the sample prep or the sample lysis method. It really starts there. And if the sample lysis method is not up to par in efficiency and in lysisability, then everything downstream is in question. And so I call it semi-automation, right? Not full automation. We're not putting the sample in and coming back and, our downstream data is completely ready for us. But semi-automation when it comes to switching from mortar and pestle to using something like a bead-rupture elite to process 24 2 mil tubes simultaneously. They're all on the same carriage. They're all being processed at 4 meters per second for 30 seconds. After those 30 seconds, I'm taking the carriage off. I'm pipetting into my downstream. I'm centrifuging the samples. I'm going into a DNA extraction, sort of walking through that efficiency that needs to happen on the front end. I think is something that lab managers and PIs need to have on the forefront of their minds, because we're often quick to look at the data analysis equipment. Scientists are using really, really fancy sequencers and quantifiers and things to analyze different molecules and proteins, but we're using a mortar and pestle on the front end. Like, do you kind of see the the gap there in the analytical nature of the instruments, right? So scaling up on the front end when it comes to the efficiency of the homogenizer and of the lysis method, I think is something that can really just streamline that efficiency and really shorten the amount of time that it takes for the customer to lyse the samples as well.
One last note on this, speaking about the bead rupture elite specifically. So talk to me about preset protocols and how those might help me get going with new people.
Yeah, great question. It's quite literally a plug and play. It's no different than, walking up to your coffee maker and hitting brew, right? It knows what to do, and you come back and you have brewed coffee. Having a preset protocol is sort of like that, right? It's a protocol that someone has validated in the lab, or that the lab has gotten from Omni and our applications team, and that they have verified that it works and it does what it needs to do when it comes to this tissue sample or this plant sample or whatever it is. So adding that protocol to the bead rupture elite is a quick pull. It's a site in the methods as well. So if you have a new tech coming in and they don't know, up from down when it comes to this lab, they're able to reference that protocol and know that, oh, I can walk up to the bead rupture elite. I can press, bacteria sample 1 and hit that protocol, and it's going to process those samples in the way that the lab has validated and verified. And it's just a quick, it makes it easy, it makes it mindless, and it also gives the lab something to reference when it comes to protocols and SOPs, which I think is really great.
Let's assume for a moment I already have some sort of benchtop homogenizer. Why do you think I would want to invest in switching over to the elite over most, pretty much all the other pieces of equipment that are out there?
Yeah, I think with the elite, you're not just getting an instrument. We don't just sell you the piece of equipment or the homogenizer, and then, it's up to you to figure out the rest. When you purchase from Omni, you get the full support and full advice of the applications team. And where we are experts in sample lysis and in homogenization, it might seem simple to the customer. I'm just, lysing my sample. What can be so difficult about that? There's literally an entire science behind it. And our applications team, myself, Caleb, Amna, Rodney, we quite literally are devoted in this job to coming up with solutions and helping those customers out, because purchasing from Omni, again, you get the full support of our applications team, and we quite literally will receive samples from you in your lab and help create a protocol in our lab that helps to homogenize those samples and get them ready for the downstream steps. So I think, again, it's not just the instrument, because, we've sort of covered our bases there. Our elite is, it's robust, it's efficient, it has a extremely robust design. The motor that's inside it's designed to run for hours and hours and hours. There's no cool down times, and all of those bases are covered. And I feel like those are, those are things that are requirements, and those are things that cannot be overlooked. If you have an instrument that doesn't have those boxes checked, then that leaves the customer with a lack of trust. And that's something that we try to definitely instill. And again, providing the application support, providing the full support of the Omni team when it comes to sales, product management, application science. We really have our customers back, and we're here to do anything we can to help streamline their sample homogenization.
All right, Gabby, thank you so much. It was great talking to you today.
Thank you, Bill. It was nice to chat with you as well.